It's my first talk in a plenary session, so I will try to do my best, not keeping you for too long, knowing that you all want to go enjoy the lunch and the pétanque that are waiting for you after my talk. Actually, this is my emotional chart over the last few months. So this is me. When I received the acceptation email for my abstract, I was like, yes, this is good news. I can present the results I worked so hard for. This is great. But then I realized what kind of session it really was. Not the type of presentation that I'm used to do, but a sort of scientific one-woman show without desk or slides. So I had to think a lot. Think about how to present my work, making it easy to understand without being boring, and how not to be too stressed. And finally, this is me right now talking in front of you with all my mixed feelings. So what I'm going to talk about for the next 10 minutes is hemophilia A, and especially genetics of hemophilia A, and the importance of using the highly effective molecular tools that have been developed since a couple of years to help us understand more about the causality of variants in regions of the genes that were not studied before, like the introns. I realized when people ask me on what I'm working on that it's not easy to explain my fascination for hemophilia. But I am convinced that we all have to keep trying to explain the mechanism of this terrible disease. As you know, hemophilia A is a constitutional bleeding disease caused by a coagulation protein defect, the factor VIII. It can be a synthesis defect or a functional defect due to the presence of pathogenic variants in the F8 gene, which spans 26 exons on 186 kilobase. And it's important to identify the molecular defect responsible for the disease because it helps for the diagnosis optimization and the characterization of carriers. But the issue we have is that despite an exhaustive screening of the full coding region of the gene, we still have 5% of hemophilia A patients that don't have any identified variant. So I just want to remind you this because it will help for the understanding later. Very briefly and easy, a gene is composed of exons and introns. And after the transcription, there is a splicing step where the introns are eliminated. And only the exons remain in the mRNA before the protein translation. So until recently, we all thought that introns were kind of useless because they are not studied, because they are eliminated before the translation, so they were not studied in the research of disease-causing variants. But in the last few years, some teams have begun to study these introns, and they discovered that variants deeply located in these introns may finally cause hemophilia A by altering all these splicing process. And this work can be done through next-generation sequencing that has provided significant improvements for a complete F8 gene analysis. So the work I'm going to present was mostly realized during the year I spent in Canada in the team of David Lillicrap. Even if my team members tried to distract me a lot with some hockey games and poutine tasting, I have to admit that I was way more excited by how to characterize intronic variants in our patient from France and from Canada with no identified variants using a NGS whole gene sequencing method. So how did we do that? First, for next-generation sequencing, we wanted to amplify the total F8 gene directly with long-range PCR and directly sequence the products. But after 3 or 4 months of optimization, it was hard to, these products were hard to amplify, and we were afraid of not have enough DNA material and not enough time for this, so we decided to give up this method, start all over, so it was very disappointing for me, but it's also as works research, and choose another NGS method. So we use a capture method. For that, the first step is to create a probe design. And each probe will hybridize on a small part of the gene until recovering the whole gene, exons in introns. We developed a small panel with both Whole F8 and von Willebrand factor genes, which represent around 400 kb. We studied 20 patients, 14 from France, 6 from Canada, having severe, moderate, and mild hemophilia A, with no variant in their F8 sequence. And after the sequencing and the bioinformatic analysis, we obtained 1,000 variants for each patient that we had to filter with different criteria. We also use Alamut software that can predict the changes on the splice site that the variant can cause. So this is for the sequencing part, but NGS is not sufficient. When you find candidate variant, you have to prove the pathogenic effect with functional experiments, like the mRNA analysis. For that, you need RNA sample from your patient, from a simple blood extraction. You reverse transcript this RNA into cDNA, and you sequence the cDNA. cDNA is supposed to be the succession of exons that I told you before. And what you hope to find is an aberrant insertion of an intronic sequence, so absolutely not supposed to be here, between two exons of the cDNA. And this insertion should be caused by the intronic variant you found. This technique is very difficult too, because aberrant transcripts are very sneaky, they are very unstable, so they hide a lot. You can very easily miss them, and you have to do the experiments several times to be sure of your results. So now that we have the techniques, let's get with the results, starting with the NGS results. After all the filtering and analyzing process, we finally get 16 deep intronic variants. When you find potentially interesting variants, you can classify them from likely benign to likely pathogenic, depending on their in silico prediction and the complementary analysis. In Canada, when I was trying to survive with the minus 30 degrees outside, because this is survival, I can let you imagine my excitation, I could even say my relief, when we found that 4 of these variants were likely pathogenic after doing our functional assays. The first 2 variants you see correspond to 2 patients, and they had in their cDNA sequence that kind of aberrant intronic insertion I told you before. So a sequence that is not supposed to be here between 2 exons of the cDNA, caused by a strong modification of the splice site by the deep intronic variant, which is located at the exact same localization where come from the sequence inserted. And during the translation, the insertion of this sequence is causing a frame shift and the apparition of the stop codon, consequently a truncated, non-functional protein. It may seem unbelievable that a variant located 8,500 bases far from the exon may affect the protein, but I can assure you it's true. This anomaly doesn't concern all the transcript. The patient is still synthesizing normal F8 with normal transcript. That's why he has a moderate or a mild phenotype and not severe. The third variant you see leads to exon skipping, and the last variant was already described in the literature. We also found 5 variants of uncertain significance. Why do we say uncertain? Because it may be that the modification of the splice site is not that strong, and simply our functional assays are still in progress, so we don't have confirmed results yet. It may also be variants that are apparently not pathogenic themselves, but we saw that they are often associated with the disease and with a more pathogenic variant responsible for the disease. We also found 7 variants that were finally likely benign. so with a few or no change in the splice site and no functional effect. Finally, and unfortunately, we still have 5 patients with no identified variant in their sequence. One last result that I want to tell you was totally unexpected. And it shows the importance of sequencing F8 and von Willebrand factor genes at the same time. This result concerns a patient that were diagnosed years ago as a moderate hemophilia A patient with no identified variant in his F8 sequence. And we found after the sequencing 2 exonic variants in his VWF sequence. Variants described as causing von Willebrand disease. So we were totally confused, and after verification, we realized that this patient was completely misdiagnosed. He's not a hemophiliac patient at all, but he has von Willebrand disease. So thanks to this study, the patient will now have a better care. So what I really want you to remember from my talk is that these results strongly corroborate that hemophilia A can be caused by deep intronic variants, so variants in regions of the gene that were absolutely never studied until very recently. Introns represent 95% of the F8 gene, so it's far more work than just studying the exons. Finally, these results lead us to ask ourselves, when does a negative result is really negative? Did we use a good technique? Did we interpret the data correctly? And can we still go far beyond the gene to find what caused the disease? That playground seems infinite. Thank you for your attention.